today. I'm really pleased today to introduce Juan Manuel Vazquez. He goes by Manny. I understand. I just met him a few minutes ago. It was a pleasure to have a few moments to talk with him. Manny's a postdoc at UC Berkeley, and he studies the evolution of longevity and health span in exceptional animals such as bats and elephants and whales. And his current research is on the role of genomic stability and longevity differences between species and the feedback loops between chromatin organization and lifespan. And as I mentioned, we have a committee that decides on the actual events that are hosted here. And I propose that we invite Manny based on an eLife paper he published recently. That was one of the two papers that was shared as part of the this event and your invitation to participate. And I still remember reading this paper. I thought it was such a great paper. In it, they introduce they investigate Peto's paradox, which we'll be hearing more about soon, and we've heard about previously in Club EvMed. I see Amy Body's on and some others on that conversation. And so you can go in our archives and hear about Peto's paradox from some other perspectives. This Peto's paradox, as I think most of you know, is this expected association we would find between body mass and cancer rates, or life history traits and cancer rates. And these associations aren't found. So if you look across species, these associations are not found between body mass and cancer rates. And this is a paradox. Why does this not exist? And the obvious resolution of this is that there are that larger animals evolve better cancer suppression mechanisms. But in this paper, they really take some novel dimensions to this question. And I'll just highlight three of them. And I think we're going to hear about this and some of the other work that Manny's working on. But they honed in on an area of the phylogeny where there's rapid evolutionary change in body mass and life history. So I really like how they're focusing in on one particular section of the tree. And they have a very interesting and powerful comparative sample because of that, including using ancient DNA, using some extinct organisms, which I think is really fascinating as well. And then their findings actually question causality. So is it really that large body mass leads to cancer suppression, or do some of the cancer suppression mechanisms actually enable the evolution of larger body mass? And so I found it just such an interesting paper in so many ways. And there's another paper that's shared as well. That I thought this would be a great topic, and I was thrilled when the committee approved this nomination. And so I'm just thrilled to have you here, Manny, and to hear more about this work and other things you're working on. So I'll turn things over to you. Thanks for joining us.
Thank you so much, Charlie, for the introduction. So just give me 1 second to share the presentation, and there we go. So I really can't say most things better than he just did. So I guess I'm just going to go straight into it. There we go. So this paper was a collaboration between me and my PhD advisor, who's here on this call, Vincent Lynch, who's now currently at the University of Buffalo, SUNY Buffalo. And originally it was at the Human Genetics Department at the University of Chicago during my thesis. Now, as Charlie mentioned, I'm a postdoc here at UC Berkeley studying different bats and kind of the same Peto's paradox paradigm, but also associations with lifespan in these different species. And if anything here catches your eye, and if there's anything that you really want to go more into, or you want to call me out on something, I want to point out that the manuscript actually is fully reproducible here on GitHub at docmanny/atlantogenata gene duplication. And this is also found in the manuscript, the link to this. And so with that, I want to then start the story. And so our story today starts with cancer, and all cancer starts with a single cell. So as you all know, our bodies are made out of cells, which over our lifetime will accumulate mutations to then become cancerous, at which point then those begin to divide uncontrollably. And this multi-stage model of cancer has been previously described in literature, starting with Armitage and Dahl, 1954, moving onwards then up to the present day, when we're talking about how we really think about cancer, not just within humans, but within different species. But for the purposes of our talk, this really boils down to two things that influence your cancer risk: the number of cells in your body and the amount of time that you have to actually accumulate cancerous mutations. So if we begin by just thinking about the number of cells in one's body, this is fairly straightforward. All cells of the same cell type are constrained by their function in terms of their size. So the cells of like liver cells in humans are roughly about the same size as a liver cell in a mouse and roughly about the same size as a liver cell in an elephant. So if you think about then within two different people, the taller person then doesn't have bigger cells, they just have more of them. And therefore, over every unit of time, every time there's a mutation happens across the different cells, there's more likelihood that any one cell in the tall person will develop a mutation that then over time becomes a series of mutations leading to this formation of a cancerous tumor in their body. And we can actually see that in real life data. So this is a meta-analysis of various different types of cancers that was published in the early 2018. And so you can see here that for all different types of cancers except mouth cancers and cancers more environmentally affected, there's a strong correlation between body size and lifespan, where the taller you are, the more likely you are to actually develop cancer. Additionally, there's also that time factor I mentioned. And so this is similarly intuitive. If you have, you're born at birth, you're kind of like a fresh, clean slate. And then over time, you'll accumulate all the different mutations that ends up causing cancer. And so if you die at the age of 30, you're not likely to be dying of cancer. But if you start living up to like 60, 70 plus, the incidence rates of cancer in the population begin to increase as a function of the population's age, indicating that the longer you live, the more time you have to accumulate mutations to actually develop cancer. So these two things, the increases in body size and increases in lifespan, are known to hold in humans, and they're also known to hold in different animals like elephants, whales, cats, dogs, what have you. But then the question is, what happens if you start comparing between species? And so this is interesting because mammals show this giant variety in body size and lifespan. So shown here is a graph of all the different mammals that we have information from the ANAGE database at the Human Aging Genomics Resources websites. And it's body size on log scale on the x-axis and lifespan on the log scale on the y-axis. As you can see here, some of the smaller animals like bats and rats, they all are on the left side of the scale. Then you have dogs, humans being an outlier, and elephants and whales. And as you progress more and more towards the right side of this graph, and as you go more and more towards the top part of this graph, you expect to see that cancer rates should be increasing then, and therefore elephants and whales should have an enormously higher risk of cancer compared to mice and bats. And so if you were then to look at that data in real life, you would expect to see, if you looked at, for example, necropsies with tumors in them as kind of a proxy for your species' cancer risk, and you were to plot that against the body size and lifespan of the animals, you would expect to see a graph like this, right? Where the elephants and the whales have a higher risk of cancer than the bats and the rats. But what you actually see from data, this is data from Abiglen et al. 2015, but there was even more recent study, including more animals and more species and more individuals, that confirms this. There is no correlation between the species cancer risk as determined by percent necropsies of tumors and their body size and lifespan. In other words, this relationship that we see between body size and lifespan within species does not hold across species. And this has become known as Peto's paradox. And so the kind of intuitive answer is, well, evolution did something, obviously, to resolve and a cancer risk of an individual, of the species, when they evolve large body sizes and long lifespans. But it's still an unknown as to how this actually occurs. However, we have tumor suppressor genes in the body, and so it's pretty easy, but we don't have to reinvent any wheels, we can just improve them. And so the nice thing is that there's two ways this can go about, either by sequence evolution or by gene duplication. And so we either enhance the pre-existing tumor suppressor genes we have, and you can usually detect this using positive selection, et cetera, or you can just make more wheels if you need to carry a heavier load. And so you can just duplicate pre-existing genes. And this also invites opportunities for other cool things such as neofunctionalization, subfunctionalization of tumor suppressors. For the purposes of this study and also for this talk, I'm actually only going to be focusing on gene duplication, but there is a bunch of other interesting things in literature about sequence evolution and positive selection in tumor suppressor genes across species if you're interested. And so now I'm going to get into the more nitty gritty aspects of the paper, of the eLife paper that was mentioned earlier. So this really gets started because we're really interested in our lab in elephants. The lab had previously discovered that elephants had 20 copies of P53 in their genome. And this led to a greater question when I joined the lab about, well, are these tumor duplications like kind of like flukes, or is this like an actual like result of a concerted like evolutionary efforts to increase the number of tumor suppressor duplications, or is it just that everything gets duplicated all the time and we have no idea, like it just so happens that like so evolution selected for like conserving all these like tumor suppressor duplications, right? And so there are three main members that we focus on in this panungulate clade, which are the elephants, the manatees, and the rock hyrax. And so these elephants are huge compared to the manatee and the rock hyrax. It was a relatively recent increase in body size. But even then, manatees are also relatively large compared to rock hyraxes. And these species are all large compared to the rest of the tree. So this unlabeled group here is the aardvark. And then you also have other species like Cape golden mole, Cape tenric, Cape golden shrew, and the tenric, lesser hedgehog tenric. And then over here in Xenarthra, you have sloths and armadillos. So as you can see, it's a very small family, and elephants are kind of like the literal elephant in the room. But it wasn't always like that. This monstrosity is the Titanohyrax, and it's the fossil ancestor of the current tiny rock hyrax. And this other monstrosity is the giant sloth that was found, like that's found in like the Americas. And it's the ancestor of the current tiny ground sloth, like tiny like three-toed sloth. So the big question then is this, how exceptional is this, right? It seems like elephants actually are no longer an exception of being the giant one in the room. It's actually, historically speaking, maybe they were all giant. So we used this program called Stable Traits, as well as several like time-calibrated phylogenies of different Afrotherian species, and also included fossil records, which was for different body sizes in the like fossil record, not only for elephants, but throughout this tree. And so what we found was here's like the phylogeny, but now rescaled for body size changes, rates of change, sorry, and colored by body size of the node. And so what you can see is that the common ancestor actually of all elephants, Proboscideans, as well as the common ancestor for the small animals like over here, were all tiny, which is really interesting because then you can see that there was suddenly huge rates of change, like suddenly big changes between a tiny ancestor of the elephant and a large like current elephant, right? And there were other interesting things, which is that Afrotheria, which is the common ancestor of elephants and manatees, actually increased in body size rapidly relative to their ancestor. And so it was not only just the elephants that had increased, but it was the manatees also that had increased, whose ancestor had increased in size. And this makes sense if you think about giant dugongs, which I didn't show here in the image, but we did talk about in the paper. There were some dugongs that were about the sizes of rhinoceroses in the literature. Stellar sea cow is the name of the species, which have since gone extinct. So what this tree suggests is that there were various body size increases that happened early on, and then suddenly elephants became small again, and then re-expanded in size much more recently in history. And this is interesting because then you would predict to see, based on this, that there should be pretty common tumor suppressor duplications, especially in the ancestral lineage of panungulates and Afrotheria. And then you would also see like another wave like that's more recent around Proboscideans like expansion. But that's not the only thing that we were interested in, right? So just because we've shown that body size increases doesn't mean that we can show that cancer risk increases, right? And that's really like the more key thing here. So there's been a lot of like math done in the literature, which I'm not going to get into the great details about how cancer risk and body size and lifespan are related to each other. But the equation boils down to this one shown right here. K being your cancer risk of an individual over the course of its lifetime before age T. T being then the age at which you're kind of taking this assessment. For example, if this is the lifetime cancer risk, then T is the lifespan of that individual, and D is the body size of that individual. And C is just a proportionality constant between body size, lifespan, and cancer risk, right? And so this is pretty clear, and there's been a lot of talk in the literature about like, this relationship. And what we did in the paper that was really interesting was that we then took, like, let's call K2 the cancer risk of an individual, for example, myself. And then let's call K1 the cancer risk of several, like my last common ancestor or someone else, right? I can then assess whether or not my cancer risk has increased or decreased by just getting a ratio between the two of them, right? And if we assume that C doesn't change significantly over evolutionary time, and over the evolutionary time frame that we're talking about here, then C cancels out. And you end up with this really simple and elegant relationship between cancer risk of different nodes in a tree, for example, that's just based on body size and body size and lifespan. So as a cartoon example, say you have this trio where the two outgroup species are like small, and then suddenly you had like a large species. You can see here and kind of intuitively that the cancer risk should be greater for this individual compared to the common ancestor that was likely small given the tree shown here. But that's only one half of the equation. There's also the lifespan aspect. And so luckily for us, one of the biggest predictors of lifespan is body size. And so we were actually able to use do ancestral lifespan reconstruction via PGLS here by using the lifespans of existing members of the tree in the phylogeny of Atlanta genata, and then just using that to create a PGLS model, which is a phylogenetic generalized least squares regression that then takes into account the phylogeny and how the different species are related, and then models lifespan as a function of the phylogeny plus body size. And so this lets us actually capture the predicted lifespans, right, of not only extant members of the tree, but also of members of the tree that we don't have lifespan information of based on their known body sizes, and also lets us predict fairly, well, who knows about accurately, but it lets us have a very precise measurement of lifespan at areas of the tree where we have fossil records for. So we have internal nodes where we have pretty good estimates of their body size. And this also assumes, which is possibly important, that the tree is known. So there's no ambiguity in the tree. So we trained this using extant body sizes and lifespans, extrapolated to the internal nodes. And then the new term I'm going to introduce to you guys is the relative incidence of cancer risk, which is just this log2 of the more recent node over the ancestral node, which just indicates basically like whether or not, like what was the fold increase or decrease in cancer risk from one node to the other. And so what you can see here is on the left is that same tree that I've been showing for the changes in body size as a function over evolutionary time. And on the right is the cancer risk as increases and decreases over evolutionary time. So you can see is that just right off the bat, if you look off the elephants, small changes in body size have a kind of knock-on effect to have like an even larger increase in cancer risk. And that's mostly because of this compounding effect of like body size and lifespan. But also from parts of the tree where the lifespan and the body size weren't coupled, we were actually able to, PGLS was actually able to kind of do that correction for us. And so at parts of the tree where there's like small, long-lived individuals, it actually does, we have a much more accurate representation of cancer risk than if we use body size alone. And so what you can see here is that dwarf elephants actually, in their decrease in size, had a increase in in cancer risk that comes from just the fact that they no longer need to select for it, right? So if you think about it a different way, they had a decrease in cancer risk that comes from their body size. In the paper, we go into talking about how really, if you think about it, it's a decrease in cancer risk whenever you think it's an increase in body size, because the animal's still alive and well nowadays, right? So if you think about it, it means that this is the compensatory decrease in bodies in cancer risk that has to come with the increase in lifespan and body size. All right. So you can see here that some the elephants generally have a decrease in cancer risk that comes with their increase in body size. That's more so than in the other nodes, which they kind of stayed relatively constant in body size and lifespan, down here. And just shown a different way here with the different actual estimates. You can see that armadillo, for example, has a very large decrease. Xenarthra had a very large decrease in body size and cancer risk because the ancestor of all xenarthrans was very large. That's the giant armadillo, the giant sloth that we saw, and some other like large armadillos in the fossil record. But pseudoungulates, so in other words, a common ancestor of the panungulates, elephants, manatees, and hyraxes, and also the aardvark, they actually had one of the largest increases in body sizes on the tree that we saw, and they had one of the largest decreases in cancer risk that we saw as well. So this kind of gives us an estimate then of where we should be seeing these gene duplications. If we predict that tumor suppressor gene duplications should be evolving to decrease this cancer risk, these purpler nodes are the ones that we would be expecting to see them appear at. So now we get to the big question. And so luckily for us, we do have genomes for various members of each of these clades, specifically the ones I'm showing here in this image, which are found all over the tree. And we also, as mentioned, have a bunch of ancient elephant genomes. So we have genomes that were generated for the mastodon, for the Columbian mammoth, for the woolly mammoth, and various other elephantids, which then allows us to actually kind of refine our search for gene duplications by looking into these like ancient genomes and seeing like what's the copy number of all these genes in those genomes. And for those who may not be familiar, we use a method called reciprocal best hit black to identify homologous genes between the human coding protein sets and all these other species. And the way that this works is take your favorite gene, and your favorite gene will have a protein. Maybe in this case we'll have a protein, and so you can then use BLAST, this BLAST-like alignment tool, to search in a different species genome for all of the instances where this like protein exists. However, it could be, for example, that you're searching another genome for P53, and so you're not going to only find P53, you're going to find P53 and the P53 family, so P63 and 73 as well are going to come up as hits. So in order to determine which of those are the real thing versus which ones are just like distantly related like homologs, you then take all the forward hits and you search back the original genome for them. And if and only if you end up the top hit on the reverse search is the same thing you started with, well, you call that a copy of the gene. So as an example, for the elephants, we had searched P53 in the elephant genome, found like 25 copies in the elephant genome. Then for each of those 25s, we took that sequence and we searched the human genome again. And for 20 out of those 25, the top hit was P53 again, indicating that 20 out of those 25 were P53 and the other 5 were just some other like spurious hits. So I developed a script to automate all this, a pipeline to automate all this, which is found on my GitHub websites in SMRec search, but also as part of the manuscript for this paper. And what we did was we automated this search for like 20,000 human coding, like protein coding genes in humans, and to look at just generally speaking, what does the landscape look like in terms of gene duplications in all these species. And so we identified from my initial search pool, 13,725 genes across all the different genomes that were found in all the different genomes. And the gene duplication rates for the pseudoungulate, so everything, all thing over here, I need to actually, there we go. For everything over here is about 9%. And outside of ungulata, so this clade over here, it was about 18 to 20%. And this is in part due to a problem of the actual genome quality. So for many of these species down here, the genomes are lower quality. They were generated in the first batch of 29 genomes and by the Broad Institutes during the advent of the genomic era. And then for the elephants, they have chromosome level genomes, and as well as manatee, hyrax, and aardvark have much more advanced modern genomes. So over here is kind of the scale, a tiny version of the body size tree that we have been showing before. And here it's just a cladogram showing the different genes that duplicated at each step. So for example, from the common ancestor of all atlantogenodons, 9 genes that duplicate in pseudoungulata, 28 into panungulata. And you can see that there seems to be this correlation between body size and different genes duplicating, but kind of sort of not really, right? it's not terribly interesting that we just find that genes duplicate throughout the tree. The real question is, what are they duplicated in, right? And so this is all the different species that we have genomes for, and this is just a simple histogram of the pathways that the genes are enriched in. So if you just do GoTerm enrichment using reactome pathway to like a database in order to see what pathways the duplicated genes participate in, what you can see is that for all all across all species, there are different genes that are associated in cancer pathways versus other pathways. And for the aardvark, it's like almost all the reactome pathways they're duplicated in, as well as TENREC. But as a proportion of the total, in all the elephant genomes and in general in all of the pedunculate genomes, the cancer pathways are more highly, like the genes are more highly enriched in cancer pathways than they are overall in other pathways. And now if you kind of look at this upset plot, which is a plot that shows basically like the different sets, very complicated Venn diagram. I'm going to highlight here the purple dots, so everything that's in Proboscideans, everything that's in Afrotheria, and then this first column here being everything, all genes in Proboscideans and Proboscideans alone. And here, this second column is anything in the African elephant in Afrotheria, so African elephant and any of the other elephants plus manatee. You can basically see that there's tumor suppression duplicates occur all over the place, but they're mainly focused actually in the elephants and in things involved like in species within Afrotheria and Panumviolata. And so based on this color coding scheme, we took the genes that were present in any of these dots, and we looked to see what unique tumor suppressor pathways were there. And what you can see is there are a lot of unique tumor suppressor pathways that are found in elephants and elephants alone, and also found in the different lineages, including elephants, where we did see those increases in body size. So this is things like regulation of P10 gene transcription, cellular senescence, which are genes that were duplicated in Afrotheria and are present in all Afrotherians. And then a lot of DNA damage signaling and specifically base excision repair, which is duplicated uniquely in elephants. So one thing is that we find all these genes and duplications, but it could just be that we're finding a graveyard of tumor suppressor genes that had been duplicated just due to random happenstance and drift, and they're not functional nowadays. And that wouldn't be interesting either. So in order to see whether or not they're actually functional, and we're looking to see if they're expressed. And so we took RNA-seq data from in vivo elephants, and we just kind of intersected it for gene duplications to see what's expressed. And what you can see is this is just a small subset of all the tumor suppressor genes in the elephant, which are expressed, but they are all involved in pretty key pathways. So orange pathways are ones that are involved in DNA damage, repair and signaling, and also like cell cycle arrest. You have others like P53, PRDX2, PRDX1, sorry, COX1, which are just genes involved in like DNA damage repair and also like cell cycle arrest and other things along that sort. And then you have a lot of apoptosis genes like Cas9, CD14, and this HTMB2, which are also duplicated. And you even have lifespan associated genes like LAMTOR5, like here, which is duplicated and also expressed. And so this is interesting because if you then go ahead over here and you look to see which one of these genes are duplicated when, a lot of these genes, when you see like something like this, where like it duplicated seemingly independently in the elephants, it's much more likely to happen in the ancestor, but one of the mammoths doesn't have it in their genome because they're ancient DNA. So it looks to be that all these genes duplicated pretty early on in the advent of the elephants, and they're still conserved and expressed in the African savanna elephants, which is the main elephant genome that we have. So in conclusion, we have the fact that body size and cancer risk increases throughout Atlantogenata, but especially increases in the Afrotherian lineage over here, and it also increases generally throughout the yellow lineages that I'm showing here. And while gene duplications are common, the elephant-specific lineage duplicates are especially enriched in cancer-related pathways, as opposed to all the pathways, like in other pathways, like more broadly in any of the other like highlighted clades. And finally, one thing I forgot to mention is these genes are mostly found within the P53 pathway. So for example, BRD7 is actually a downstream effector of P53 signaling, whereas not shown here, but highlight, oh, sorry, here, STK11 is actually upstream of P53, is involved in regulating P53. And so this is all interesting because it indicates that sometime we know that the P53 duplication occurred roughly at the start of all peduncular, it occurred in Afrotheria. And so it indicates that there's a combination of some genes which duplicated beforehand, which were involved in P53 regulation, and some genes that duplicated after P53 that regulated, which are downstream effectors. And so it's very interesting because it suggests then that not only the P53 duplicates, and that was already important, we knew, for tumor suppression, it also indicates that other genes duplicated before and after to A, lower the toxicity of P53 possibly, and then B, also increase and specialize the different downstream effects when P53 is activated. So this is only the beginning. So I also, for my thesis, had done like this same study, but for all these other different species. So we are currently working on Riker and evolutionary and the evolution of that proportionality constant C that I talked about earlier across all the different vertebrates for which we have cancer risk data or just body size in terms of for Riker. And also as part of my current work here in Berkeley, I'm sequencing 9 novel meiosis genomes here in bats to study evolution of longevity and longevity associated traits by focusing on the 3rd longest lived bat, which is the most common bat in North America, the little brown bat, and all of its closely related species found on the West Coast. And finally, one thing that we've had our eyes on for a while is doing a massively parallel assay of tumorigenesis and tumor suppression by just knocking things down and using single cell assays of tumorigenesis, et cetera, to actually see how they function in vivo on like one fell experiment, right? So at this point, and actually generally, I see a lot of questions in the chat that we'll get into, but I had left this question open as kind of like to direct all the future discussion, which is that gene duplication is intrinsically thought of to be a kind of possibly toxic thing, right? So you suddenly increase the dosage of this gene, and now you're asking the question of, well, is it good or bad for the animal? But in this case, we're talking about genes that are pro-apoptotic and anti-growth. And it's known already in the literature that for some of these genes, for example, overexpression of P53 leads to renal failure and developmental defects. But in the paper that you guys read as well from Garcia Cow, what they did was they did a transgene that has all the response, regulatory elements included. And so those mice were healthy and tumor resistant. And that would more closely mimic a whole locus duplication of P53. On the other hand, you have other tumor suppressors like RB, for example, which if you overexpress it, regardless of if you overexpress it just in terms of raw copy number with high efficiency like promoters, or if you use a TET regulated promoter, you always see this relationship where the size of the mice is impacted negatively by the amount of RB dosage. And another tumor suppressor I found in the literature, ARH1, if you overexpress it, also causes a reduction in body size alongside the tumor resistance. So this is really interesting because you'd start seeing this pattern. You start wondering how in the world elephants and whales actually get to be the body size that they are, right? Because they're overexpressing a lot of these things, and yet somehow they're not becoming dwarf mice, right? And then there's some other genes, which, like, for example, the BRCA gene family. In the literature, I've tried to see if people report dwarf mice with these, but I can't find anything. And that's one of the big problems of you just do something and you think that the cool thing is X, but actually the cool thing is Y, but you didn't publish Y. So now I have to wonder why. And also INK4 ARF locus is really important in inducing cellular senescence and inhibiting cancer in humans. And it's known that if you overexpress these genes in cells, you get induction of senescence. But yet there's no reported body size changes in these mice when they do these constitutively overexpressed lines. So all this together indicates that there's a lot more to this story. And I'm hoping to over the course of this year, actually finish up a review, just kind of talking about all this. But at this point, I've talked enough, and it would be nice to actually have a discussion.
Thanks, Manny. That was fantastic. I see some hands clapping, virtual and otherwise. Great to hear your presentation of this work. We have a lot of questions and comments in the chat. It's been pretty convenient for you to have a co-author who's responded to a lot of these.
Yeah, no, thank you so much,
questions and comments. But I think we should loop you in, and also other members of the audience who may want to ask some of their questions verbally and maybe explain some of their positions a little bit better. I was having trouble following a little bit of this. Do we have anyone who has a question to get things started?
I do see that there's actually one thing that I forgot to clarify that got asked. So that body size cancer relationship, that's actually like the meta-analysis I was looking at took studies from 6 different consortia across the world, including not just Europe and like Americas, but also in Asia. So this is something that's like it's a global phenomenon. It's not just a European diet issue.
I guess I'm happy to start off since you brought up that. First off, great talk, man. He's so well presented. It's really fantastic and great perspectives. So I'm curious about, and it goes back to some of the data that you had shown earlier, where they've correlated, body size within species with cancer risk. And I guess, I'm wondering about some of the complications because, when things correlate, it doesn't mean causation, and everyone jumps to more cell number means more cancer, but there's also, it's going to correlate with IGF signaling, growth factor signaling. And you also have to sort of think about it's almost like life history theory life history changes within a species, within a lifespan. I mean, within species, and that,
the history.
Sorry? Oh, I don't know if someone was jumping in, but basically larger individuals are sort of front loading the investment. I mean, they're going to, in fact, I'm pretty sure larger individuals just live shorter lives in general, not just from cancer. So they may even age more rapidly. So is this just sort of a shifting of the aging and cancer curves together?
So that actually gets right at the heart of that last thing I was talking about like the tumor genesis and the actual toxicity effects of some gene duplications. So IGF in receptors are also duplicated in elephants. So I'm gonna first go take a couple steps back to answer your question and then keep going. So yes, it's known that in humans, for example, individuals in populations where they have they're heterozygous for IGF, non-loss of function mutants, are long-lived, and they're also smaller, not terribly surprising. And Andre the Giant is probably the most famous example of someone who was born with an IGF mutation that caused hyperactivity and ended up living a truncated lifespan. So it's known that the IGF signaling is kind of at the crux of how you get large. But then the problem is that, either way, right, if an elephant and a whale are expressing let's say 1 million times more IGF than humans are, it's still is kind of you can still do it a proof by counter example, right? They're not dying of cancer because they're not dying at a bit at the embryogenesis states, right? So they're basically alive and well, even if it's despite of maybe this large dose of IGF signaling, which indicates that there have to be evolutionary ways of compensating for that. And so then that still kind of doesn't get at fully the whole this tug of war between the duplications of IGF genes and then some positive selection to kind of fine tune them, and then duplication of tumor suppression genes and then fine tuning them so they're not just haphazardly causing tiny kidneys everywhere, right?
No, I totally agree. That's a good reason why it's just not, one can't count cell divisions to come up with cancer risk.
Yeah.
Can I ask a question?
Go ahead, Neil.
Yeah, so I run a histocompatibility and immunogenetics lab and interact routinely with the blood and marrow transplant program as well as with solid organ related physicians. And you're averaging over every kind of tumor. And I'm wondering if you're focused solely on solid tumors, number one. And number two, if you're not, and you're including hematopoietic tumors, I wouldn't even expect there to be necessarily the same results for AML versus CML versus lymphoma versus myelodysplastic syndrome, et cetera. So, I think it's very impressive work in a lot of ways, but I'm just concerned that when you average over very different kinds of cancers, you may get averages that apply to nothing.
So there's a really interesting review from 2019 that tried to answer this question. This is not the same data as the Aberglin et al. Data. So the Aberglin et al. Data was when they did a necropsy of an animal that died for whatever reason. They looked to see if they were tumors or also leukemias. And so that's like a whole other thing. But to answer your question, there was another review that kind of like went all the way across vertebrates. So they looked to see in the literature what kinds of cancers were reported across vertebrates. And the interesting thing was different cancers correlate differently in body size and lifespan, like with body size and lifespan. So leukemias tend to be kind of neutral. So and it kind of makes sense, right? It's like all organisms like will have blood, and blood does scale with body size, but the number of like leukocytes in the blood, etc., doesn't necessarily scale with the body size, right? That scales probably more things like environment and such. But yeah, there is a difference between like different types of cancer scaling differently with like body size and lifespan. That was something that was shown in like the first figure where like not all cancers were slightly associated, but some of them were extraordinarily associated with body size and lifespan. But nonetheless, it's still like a general trend. So we weren't really averaging like everything, but we were just kind of like observing that like everything had some effect of body size and lifespan within a species. This is also very different from across species, where like across species, there's just no correlation for any type of cancer. And that is the more recent Aberglin 2020, I believe, paper.
Okay, thanks.
I have sort of a crazy question, Manny. There are these infectious cancers like in the Tasmanian devil, and there's a dog venereal cancer that's transmitted sexually, obviously by the name, as you can tell. And when I look at that phylogeny, I see there's kind of clusters of these duplications in places that aren't necessarily associated with increases in body size, with increases in predicted risk. And I'm just wondering, could there have been kind of outbreaks of these infectious cancers occurring throughout the tree of life? And do we have any, maybe Amy or somebody else has thoughts on this too. Do we have any expectations for where that would occur, what lineages that would occur in? I mean, socially, obviously you have to have a lot of, contact between individuals. So maybe there's a correlate there. And also, I mean, just thinking about it from your side, Manny, I mean, are there any kind of signatures that would reveal, either in terms of particular genes that would be duplicated, any kind of particular defenses, or because it could come on so suddenly, would it be a different kind of selective force that might result in some kind of genomic signature? Sort of a crazy thought there, but I thought I'd raise it in case anybody has any thoughts on it.
I know that I think the key word that you used was contagious cancers, right? So it becomes more of an issue of self versus non-self in the Tasmanian devil. And also and in other species as well, like the prevalence of like viruses that carry like oncogenes in them. And so I think that's actually like the more like important thing is to make that distinction, because in Tasmanian devils and in cheetahs too, I think also have a high levels of abnormally high levels of cancer, although I don't remember if it's contagious. It's just an issue of like tiny inbred populations not being able to tell self and non-self apart. So the cancer can pretty easily spread. At least that's my understanding of it. I could be wrong. Generally speaking, I feel like the contagious cancers has been my I don't need to worry about you kind of deal, because it's not like straight up like body size. Yes, Amy, yeah. So like Amy kind of brought up. Do you want to actually talk more about that?
Yeah, so I guess you would maybe, by the way, great talk. I loved it, and beautiful visuals. It's fantastic. I don't know. I love this question, Charlie, because we talk about contagious cancer a lot in our group because it's fun. And I think maybe you would have to predict the genetic bottleneck within a species first. And so before you would maybe see an outbreak in contagious cancer per se. So.
Yeah, no, I would love to have nothing more and nothing less than like a giant population genetic database for every species available. So that way I could do a giant analysis about like that.
Yeah,
Um,
and the other thing is, you know, like Charlie mentioned, is you need the transmission mode, right? And so we see bivalves, actually, there's like 7 or 8 species now with contagious cancers. And so water transmission might be good. The others, bitey type animals like Tasmanian devils, you need that, or obviously through sexual transmission. And so you have to have maybe these characteristics in play for that contagious cancer to have a takeoff, right?
Yeah.
So.
Virally transmitted cancers are the ones that I find the most interesting, like amongst these, because it's like, would there be like, what would be selectively like more favorable, having like a more hyperactive immune system that identifies a variety of these, or just, you know, developing like increased cancer resistance against like viral oncogenes? And even then, like, you know, is this big enough of an issue that you even evolve anything to combat it, right? So like human papillomavirus being like one cool example about this. It's like, you know, where we were on, it doesn't kill people young enough to like actually like for us to predict there would be an evolutionary pressure against it, right? So we probably just continue to be a problem until the end of time, right?
Okay, we have, yeah, I'll turn things over to Meredith. Do you have a question from the chat you want to call on, or anybody else want to call on? Anybody else, raise your hand, please. We have a couple more minutes if anybody has a question.
Yeah, do feel free to raise your hand. If not, I'll pull one from the chat that I thought was really interesting and sparked a lot of great discussion. So Cynthia Bell had asked in the chat, within organisms, some organs grow in cell number for a span and then perhaps in cell size, others grow in number for a longer span. And how might that figure into this model? There's a lot of great discussion in the chat. I wanted to get your thoughts on it, Manny.
Actually, wait, I think Cynthia dropped off. Oh no, she's still here. Because like I'm trying to think which, I know that in Drosophila, for example, like it's the usual like case where it's like a bunch of like nuclear divisions and in some late cellularization. This is like very early on in like development. I don't know if animals that like do this like once they're like much later on, like during like adult growth, etc.
The example I was thinking of is lungs in people, where the airways grow perinatally in number, and the airways don't get much bigger after about the age of 2 or 3, yet the lungs themselves and the alveoli continue to grow into the mid-20s.
Interesting.
And so it seems as though that might help you figure into your model.
My gut instinct is the most important thing is the division, right? So like the size, not as big of a deal, because like this, a big size, so long as it's not inflammation based, right? Or something that's like is like oncogenic in of itself, it shouldn't really play too much of a factor in the cancer risk that comes with it. So if there's a lot of early on like cell divisions, if it happens in an animal where like the cell, like, you know, like the DNA repair isn't like as like stellar, and so like you're basically tripping over yourself to try to make more cells, I would assume that that would be very carcinogenic. But I don't know of an animal that like, and so like one organ, if that happens, and it was a small animal, I would assume tend to see an increase in that particular organ's like cancer risk compared to the rest of the body. So like maybe some hypothetical mouse, right, that's like small, already kind of predisposed to cancer, and then like in a sister lineage, like its lungs like are much larger, maybe because it lives in a higher elevation. So in a recent evolutionary shift, the pressure was on build bigger lungs, so you would develop more like cell divisions, and then you have this like issue where, like, you know, all else being equal, species A has more lung cells than species B, and it didn't evolve much higher, more rigorous like cell cycle controls, right? So now the result is that you'll have higher incidence of lung cancer in one versus the other.
I'm thinking about that in people, so thank you.
Thank you for that question. That was actually really, it was nice.
You're welcome.
I see Kalen has his hand raised.
Right,
Yeah,
Dave?
hi, thanks so much. That was an awesome talk. So one of the things, and Vinny kind of, addressed some of this in the chat, but one of the things that struck me is like sort of a separate paradox going on, which is gene duplications are sort of a type of mutation. And so you have these gene duplications, but they're also reducing mutation rate, presumably. So I'm wondering how you think about evolvability or the ability to continue to evolve these kinds of cancer suppressors in light of the fact that maybe they're actually reducing mutation rate or evolvability potentially.
Yeah, no, that's yet another one of the things that kept me up at night when I was a grad student. It's like a general issue with aging in general and schools of thought in aging in general of the more good you get at preventing mutations, the worse you get at evolving anything better. So it becomes kind of we go from Peto's paradox to Zeno's paradox of every time you try to get to the halfway, to the final mark, you get halfway there and you're halfway further, and eventually you're just splitting hairs. And that's kind of how I've been thinking, how I thought about all this stuff as well. If you have an increase in cancer risk, it means you're undergoing more cell divisions, which means that even for the gametes, right, you're still undergoing probably some more cell divisions than usual, than a smaller animal is. And so it becomes an issue of you're decreasing the cancer rates, your cancer risk by evolving all these tumor suppressors, et cetera, but you're also increasing your mutation rate by just having more cell divisions. So those two, it's this tug of war that you're in, this fine line you're constantly waging. And I imagine, therefore, the reason why intuitively I would expect that you would always have body size preceding the tumor suppression duplication, even though that's not actually what we always see in the data. Is this thing of you increase the mutation rates, and that allows for more evolvability of these tumor suppression duplications, et cetera. But I think one thing I didn't talk about, but I alluded to, was regulation plays another important role. And Amy actually mentioned in the chat right now, tissue-specific cancer defenses. And I think that that's actually the more important part here, because if you can evolve a way of reducing your tumor risk somatically or in individual soma versus in the germline, right, then you can kind of get away from this evolvability issue, because the germline are free to experiment while the soma needs to just be kept in line until you can reproduce, right?
Thank you, that's awesome.
Okay, we're almost at the top of the hour, so we're going to wrap up here. Thank you, Manny, for a fantastic talk. That was really great. Another round of applause for Manny. And Vinny, nice of you to join. I appreciate your help in answering questions in the chat. That was really good as well. And thank you all for joining us. I want to just share with you our upcoming Club EvMeds. We have, I believe it's 3 more coming up on the schedule. Maybe there's one that's not listed here. Meredith can correct me if I'm wrong. Here's the great talk we just heard. And we have 3 more. We don't have titles. Well, we do have titles for at least one of them. We have Amanda Leah speaking on July 21st. Then we have Molly Fox on August 17th, and then Concha Cole on August 24th. And we're actively preparing for the fall semester. So please, let us know your ideas, and we'll be happy to send them to the committee and discuss them in the committee. Anyhow, thanks everyone for joining. Really great to see everyone, and hope to see you at future one of these events, and also the upcoming International Society for Evolution Medicine and Public Health Conference. Thanks everyone.
Thank you.
Thank you, Meredith.